Okay, here's what you hopefully figured out. When the A1 allele is very rare, fitness is pretty low. Mean fitness in the population is pretty low. The few A1 alleles that exist in the population have a really high fitness compared to these A2 homozygotes. So the more A1 alleles you add, the more the fitness goes up. Okay, so if you start over at this extreme, the fitness is going up starting over here. If you start over at this extreme, A1 homozygotes only have a fitness of 0.8, and every A2 allele in the population is going to be in one of these heterozygotes. So the more heterozygotes you get added to the population, the more the mean fitness of the population goes up. That means that as you're going to start around here, as you add more A2 alleles moving this direction, this is also going to go up. Oh, we've got a different situation from the last time. Here we've got a line that starts here and goes up. We've got another line that starts here and goes up. The shape of the line connecting them has to look something like this, with a maximum somewhere out here in the middle. What's going to happen due to natural selection to these allele frequencies? Well, it's what I said last time. Allele frequencies are going to evolve uphill. Okay, so natural selection is going to push the population uphill until it's at a maximum. But in this case, it doesn't mean that one of the alleles is going to be driven to extinction. If the A1 allele is very rare, natural selection will make it increase in frequency until it reaches a frequency of about 2/3. And vice versa. If the A2 allele is really rare, it's going to increase in frequency until it's at a frequency of about 1/3, and the A1 allele is at a frequency of 2/3. So no matter where you start along this line, no matter what allele frequencies you start with, natural selection is going to push you to the same endpoint, and it's right here. Neither allele goes extinct. They're both maintained at a reasonably high frequency in the population. And again, the relative frequency, or the relative fitnesses, of these three genotypes don't matter as long as the heterozygote is the most fit. You can have lots of different shapes, but the maximum is always going to be somewhere here in the middle, which means that natural selection will maintain both alleles in the population. So when the fitness of the heterozygote is higher than the fitness of either a homozygote, neither allele goes extinct. If all of this graphing and mathing is messing with your head, here's an easier way of understanding that. Remember from the Prout square, when an allele is really rare, it's found in heterozygotes. Almost entirely in heterozygotes, actually. When an allele is at a frequency of 1%, 99% of those alleles are found in heterozygotes. Well, if the heterozygote is more fit than either a homozygote, that gives that rare allele a real advantage. But you flip it around, and the other allele is the rare one. Now it's the one that's found mostly in heterozygotes, and it has an advantage. In other words, both alleles have an advantage when they are rare. If an allele has an advantage when it's rare, it's really hard to drive it extinct. Natural selection will always favor it increasing in frequency when it's rare. This is the case for a few genes that we know of in real populations. It is not very common. Usually the heterozygote is not the most fit genotype. One classic example where it is the case of the sickle cell gene. We talked about this a little bit in the lecture on mutation, because there's one mutation in a protein coding region that switches normal hemoglobin over to the sickle hemoglobin that causes this sickled shape in many red blood cells. Now, if you have two copies of this allele, your red bloods all have this really strong propensity to sickle. And that can cause very severe health problems because those sickled red blood cells end up piling up and clogging small blood vessels. It leads to a number of health problems, and usually individuals with two sickle cell alleles die much younger than people with at least one normal hemoglobin allele. However, this allele is at a pretty high frequency in some parts of the world. And those parts of the world are exactly the parts of the world where you find really high frequencies of malaria. The malarial parasite is this very odd little parasite that actually hides inside your red blood cells. Mosquitoes bite people. The parasite gets out of the mosquito's salivary glands and into the person's blood. It drills into red blood cells and hides inside them. However, if you've got just one sickle cell allele, your red blood cells have this tendency to sickle and collapse when they get invaded by a malarial parasite, and that kills the malarial parasite. But the rest of the time, the heterozygotes blood cells do not sickle and collapse. So they enjoy the health benefits of not having sickle cell disease. And they also enjoy the health benefits of not having malaria. In areas where malaria is very common, this gives heterozygotes a big edge in terms of fitness. Historically, in some regions, mortality due to malaria has been this high, something like 7%, 10%, 20%. And when that's the case, heterozygotes, individuals with one sickle cell allele, have a pretty extreme advantage in fitness over either of the homozygotes. And for that reason, in those parts of Africa where you see a lot of malaria, you'll see both the normal hemoglobin allele and the sickle cell allele maintained by natural selection at intermediate frequencies. Okay, what about a third case where the heterozygote is the least fit? I won't go through the long explanation here. You should be able to stop this video and figure out what the shape of the curve on this graph is going to look like. Go ahead and do that now.